Hello everyone. Today we will be talking about antigenic structures and biochemical test of Neisseria gonorrhoeae. If you want to know about the morphology and other characteristic features of Neisseria gonorrhoeae, you can go to my previous video. Here we will be focusing on the antigenic structures. But before starting with antigenic structures and biochemical tests, let us know few important things about Neisseria gonorrhoeae, that it predominantly infects the mucosal epithelium of human urinogenital tract. So basically, the infection and the place where it grows is a human urogenital tract, but it can also infect the exposed mucosal sites such as rectum, pharynx, and conjunctiva. How all this happens, that we are going to read in the next video when we will be talking about the pathogenicity of Neisseria gonorrhoeae. So here I just want you to keep some these things in your mind and then study the antigenic features, antigenic structures. So the places where it can infect and colonize, and it also has the ability to undergo phase and antigenic variations. What is phase and antigenic variation? That means antigenic variations give rise to expression of different kind of proteins, and the phase is like phenotypic character of the bacteria changes. So this is also found in this Neisseria gonorrhoeae. It's a very old bacteria. It was reported in 1550 BC for the first time, and still it is surviving and it is gaining resistance against many antibiotics. This is because of this feature it possesses, that it can undergo both phase and antigenic variations, and it avoids clearance by immune system. Why? Because of the variations. It keep on changing its color, its shape, and the proteins it expresses. Shape doesn't means that it is a diplococci, so it keep on changing its shape. No, it's the physical features, its phenotypic character, it keep on changing. So what are the different antigenic structures present on gonococci, that is on Neisseria gonorrhoeae? Let's sum up in this pictorial diagram, that is lipopolysaccharide, then peptidoglycan, protein 1, pili, which singular of is pilus, and pili is uh, plural. Then IgA protease is also protease enzyme is also present in this. Cytoplasmic membrane to act as a, it protects the bacteria, and its cytoplasm is also playing a role in as an antigenic property. What, how, and what they are doing. Let's go to the next slide and study. So coming to lipopolysaccharide, this I have shown you in this pictorial diagram. This you can see that it is something present on the outer membrane. So how is it working? It is it controls the bacterial entry into human mucosal cells. How it is doing this? It is stealing the celiac acid present in on the surface of immune cells. That is this Sigilex is a receptor which is going to, if celiac acid is there, it's like if this particular dress is present. For understanding, we can take an example that if you are wearing black dress, you will be allowed entry inside the cell. So what is this lipopolysaccharide doing in this case? It is just taking the celiac acid and dressing itself in this in such a way and forming the lipo-oligosaccharide molecules. Chemical reaction is taking place. I'm not going into the chemistry of it, but just to make it at this point, just to make it clear, it is like it wears a dress. That means it wears a dress of celiac acid. That means it gets salicylated and then it enters inside the neutrophils, while neutrophils are there to protect us, but their neutrophils are fooled by this, and it allows the entry of lipopolysaccharides in this. Because of this lipopolysaccharide, the Neisseria gonorrhoeae enters inside the human cell and thus fools the human immune cell, and these are the ones which are responsible, are susceptible also. So if we are going to develop any vaccine or we are going to work, then this is the part which we can target. So this is responsible for bacterial susceptibility to killing by antibodies and complement also. Then the peptidoglycan is another which I have shown in the pictorial depiction. This is the peptidoglycan fragments which are released from the cell, and they come on the surface of the cell of bacteria. And this is in the lab, it is reported to cause damage to human fallopian tube tissue, which is in the organ culture. And this whole system mimics the damage which is seen in the patients with pelvic inflammation disease. So peptidoglycan is also playing a role. Then there are three outer membrane proteins which are also responsible for colonization in the mucous membrane of human cells. And this is porin ion channel protein, that is pore B, while the colony opacity associated protein, this is also the protein responsible. These are three proteins which are responsible for colonization. Reduction modifiable protein. Reduction modifiable protein is the protein which is genetically modulating. All these proteins are responsible for genetic modulation and thus fooling the immune cells. They are changing the surface in such a way that immune cells are fooled. And two key pathogen associated molecular patterns are also present on this, which fools the immune cell, and thus bacteria gains entry inside the immune cells. Now, one more very important antigenic feature which is present and which is responsible for most of the virulence, which is the key virulence factor. This is the pili. And pili is plural and pilus is singular. And this you have seen on the first diagram, a pictorial diagram that I have presented in this video. There you can go and see the pilus. It's a short hair-like structure which facilitates adherence to mucosal surface. The adherence, that means the bacteria is continuously adhered to the mucosal membrane because of this pilus, which can extend and then contract. So extension and retraction is because of this pilus, and thus it facilitates infection as well as colonization. Now let's come to the next slide. That is IgA. I have IgA protease. I have talked on this enzyme in other. It is present in other bacteria also. So major role of this enzyme in this gonorrhoeae is also to contribute in the colonization of mucosal surfaces. Now I have told you that cytoplasm do play a role. That is, it's cytosolic glycotransferases is transferred, and which determine the glycan profile. That means that glycan profile is present on the lipo-oligosaccharide, that is lipopolysaccharide membrane, which have a molecule of lipo-oligosaccharide. When a glycan that glycan gets in this lipo-oligopolysaccharide gets the structure of glycan from this cytoplasm, and this basically this cytosolic glycotransferase enzyme. And this glycan keep on changing, the structure patterns keep on changing, and that is why this bacteria, one strain of this bacteria can show different phenotypic characters and thus can fool the innate and adaptive immunity both. Now coming to the biochemical reactions by Neisseria gonorrhoeae. On just one slide, I have told all the important, put all the important biochemical reactions that you have to remember for the test purposes. That is, it is oxidase and catalase positive. Now, what are the bacteria, why they are oxidase positive, and why catalase positive or negative? If you want to read about it, you can go to my previous videos where I have discussed why certain bacteria give oxidase test positive, while certain give negative. What is the thing that is present in bacteria which is responsible for the test to be negative or positive? So whole one video is there, so I'm not going in the details of why this bacteria is giving oxidase positive. I have just summed up here that this bacteria Neisseria gonorrhea is oxidase positive, catalase positive. It can ferment glucose and produce acid. It cannot ferment maltose. Very important character in MCQs. Lot of MCQs you will be asked why. What is the major difference between the biochemical test of Neisseria gonorrhea and Neisseria meningitis, or gonococcus and meningococcus. Otherwise, all the tests of gonococcus and meningococcus are all the biochemical tests are same, while only the difference between the two is this is maltose. Neisseria gonorrhea do not ferment maltose, so it's negative. While meningococci, that is Neisseria meningitis, ferment maltose and produce acid. Lactose is not fermented and sucrose is not fermented by meningococci as well as gonococci. So this table you can learn for meningo as well as gonococci, only differences of maltose. So just remember that Neisseria gonorrhea do not ferment maltose. So M for M, that is maltose, is fermented by meningococci. Thank you so much for patient hearing.